I'm going to emphasize why we pick these in vivo systems, why they're good for microbeam kind of studies, and the kinds of studies that have been done in the past to get the juices flowing, to get you guys thinking about various in vivo systems that you could actually use. The first one is the Medaka fish embryo. And this is being done in collaboration with Bill Dinan down in the Medical College of Georgia. And this is what the embryo looks like. It's a well-established system. It's optically clear. For microbeam studies, that's an important thing. You should be able to see what you want to look at. At stage 28, you have these two dark eyes. And right between the two dark eyes is the development of the midbrain. The egg itself is small. It's a diameter of about 1.2 millimeters. Again, this is important so that you can target things within the range that the microbeam, within the acceptable range for the microbeam. And it's also a good model for embryonic development, which is Bill Dinan's main research emphasis. So this is, again, a schematic of the Medaka egg embryo at stage 28. And to actually microbeam irradiate it, the embryo must be oriented with respect to the microbeam. So if this was the bottom of the dish, what you want to do is get these eyes down to the bottom of the dish so you can actually target the midbrain. So what we used to do was to roll this around. You put a cover slip on top of it, then use your finger to roll the egg around so that you got the eyes down at the bottom. It used to take about 10 minutes. And then once you get the eyes down, we were using a proton beam in this case, you could actually target the midbrain right between the eyes, like you remember in the picture previously. Gerhard has since developed an offset hinge where you can actually use a tracker ball to roll the egg to get in the right orientation. This is the embryo, that's the midbrain. So once it comes in position, you can hit it. And this is far easier and faster than you actually using your finger to move it around. So throughput is increased tremendously based on that one change. So this was the experimental design that we did with Bill Dinan. Antonella did with Bill Dinan. Irradiate the midbrain using 4.5 MeV protons. The beam diameter in this case was 50 microns, which is much larger than a typical microbeam, but in this case we were just trying to hit the midbrain. And rather large doses, 10,000 to 20,000 protons delivered right between the eyes, essentially. Whoops, what happened? Okay, so these are the results. This is the midbrain. These are the two eyes. The green spot here is where the beam was targeted, and these red dots here are individual cells that were apoptotic. So essentially, the experiment was you irradiate the embryo between the eyes at that particular spot. They fix the embryos and they look for signs of apoptosis. And what they found was, in this graph here, this is frequency of apoptotic cells. Where you hit, this is the controls, you get about 50 with 10,000 protons and 20,000 protons. And there was a significant difference between the controls and 20,000 protons in terms of apoptotic cells. Now the important thing here is, this is where the beam was targeted. And if you see apoptotic cells here, that's an indication of a bystander response. That is, you irradiate up there and you're seeing responses away from this site of irradiation. There was no significant difference between the controls and 10,000 particles. There was significant difference between those and 20,000 particles. Yeah, those are the p-values there. So this is one of the in vivo systems that's been used at RARAF and in conjunction with the microbeam to ask the question of, if you hit cells in one particular area, what happens cells away from the zone to cells away from the zone of irradiation. All right. Again, that's where the spot. So the conclusion that they made based on these data was that this fish embryo, the Medaka fish embryo, is a suitable model for microbeam in vivo studies, particular bystander kind of experiments. The other in vivo system that Antonello has been working on is C. elegans, and this is a collaboration with Dr. Oliver Hobart at Columbia University, Howard Hughes Medical Institute. And C. elegans is, again, I'm not sure how many of you are familiar with it. It's a worm. It's very well characterized. It's got 10,028 cells in a 184? 984 cells in the adult. That's a picture of it, what it looks like under the microscope, and that's a schematic of the various organs. Okay, and why does it make a good in vivo system for microbeam? It's small, about a 1 millimeter in length. The diameter is important. If you want to hit with a microbeam, you'd like to see particles coming out the other end. So it's 50 microns is a good size. It's a transparent body, so you can see into it. It's got a short life cycle. It takes about 3.5 days to go from a single cell, fertilized single cell, to an adult. The adult lives about, the entire lifespan is about 2 to 3 weeks. It's very well characterized, like I said. The genome has been entirely sequenced. And importantly, to make it, what's the word? Relevant, that's the word. It shares cellular and molecular structures with higher organisms. So this was the experiment, the initial experiment, using C. elegans with a heat shock protein tagged to GFP. And the heat shock protein is expressed in particular sites in the non-stressed worm. And when you stress the worm, you get an increase in expression at various other points. So this is the control. We are using protons, 3 MeV protons. In this case, the beam diameter was about a micron. And we irradiated Antonello irradiated worms with protons and then looked at the responses 24 hours post-irradiation. So this is the control. This is where the worm was irradiated, the little red star. And you can see that even though you irradiated the worm at this spot, you get expression of GFP tagged to heat shock protein 4 along the entire length of the worm at 50 protons and 75 protons at that particular site. Again, this is indicative of cellular responses away from the site of irradiation, bystander responses. This was actually published in 2009. So they got a little more creative and took it one step further, where they actually generated a new strain with dual labels. And in this case, they were using heat shock protein 4 tagged to GFP, like in the previous experiment. But then they crossed it with a red fluorescent protein tagged to DAT1, which is a plasma membrane transporter, and is expressed only in 8 neurons in the cell. So this is a picture of a crossed worm. You got heat shock protein 4 and DAT1. The DAT1 in red and heat shock protein in green at 1, 2, and 3 sites. And this is essentially as a function because of the imaging system. So the idea was, again, this is in the posterior end of the worm. The DAT1 is expressed only in this PDE neuron in the end of on the 2 sides, essentially, one on the left and one PDE right and PDE left. So, and that's, if you're looking at the section of the worm, that's where the DAT1 is expressed. So that's where we targeted, and then again looked for GFP heat shock protein 4 expression along the length of the body. So that's where the protons went in. And there were four independent experiments. Worms imaged immediately before microbeam radiation, hit with 75 protons delivered to one of these neurons, one of those red neurons. Control worms are Mach irradiated, and then were imaged, both irradiated and control worms were imaged 24 hours after microbeam irradiation, and then the GFP expression, indicative of heat shock HSP4 expression, was quantified using QuantaWorm, which Guy wrote. And this is essentially the picture of the program where it measures intensities along the length of a worm, and you get this kind of profile. And these are the results. At a control worm, you can see the 2 neurons, the DAT neurons, at time 0 and at time 24, and it doesn't change much. This is 75. This was a worm that was imaged prior to being hit with 75 protons at time 0. This is where the protons were delivered. And then you're looking at GFP expression along the length of the worm. And obviously, you can tell the difference that, even though the worm was irradiated at that point, you get enhanced GFP expression, or enhanced heat shock protein 4, away from the site of irradiation. Again, bystander response. And this is the quantitation of the results. GFP expression in fold increase in controls, and 75 alpha particles, protons, sorry. 63 worms in the controls, 45 worms in the irradiated population. And what you can see, there is a significant difference, meaning that you are seeing a bystander response in these worms, as seen by enhanced GFP or enhanced heat shock protein 4 expression away from the site of irradiation. So to conclude, the C. elegans study, microbeam irradiation with 1 micron diameter proton microbeam induced stress in unirradiated areas. And take it one step further, if you target specific neurons, you can induce both local and distal GFP overexpression. And ultimately, the goal of this whole study was to demonstrate the suitability of using this model system, the in vivo C. elegans model system, in microbeam in vivo studies. And that's essentially the bottom line, that we have the capability to target in vivo 3D systems or in vivo systems, which essentially takes cell culturing and all the information you can get from cell culturing to a whole different level. And well, Medaka and C. elegans are the two so far. We'll be developing others as well, and we're always open to new ideas and new suggestions of what we could do. And thank you, Antonella.